Welcome back. This is part 2 of chapter 4. We're going to focus on gene action from genotype to phenotype. So when we left off from part 1 of chapter 4, we were talking about how genes can lead to phenotype. And this is just sort of an extension of that, and essentially building on the idea that when genes are expressed, sometimes we get the phenotype we expect and sometimes we don't. And the reason why we might not get the phenotype that we expect is because genes do not act in isolation. And instead, things in the physical environment as well as the biological environment can influence how genes are expressed and therefore the phenotype that we're going to see. So a couple of examples, a few examples of how the physical environment can influence the phenotype. A couple of things, temperature, light, nutrition, humidity. An example of how temperature could affect the phenotype is seen in a temperature-sensitive Drosophila mutation called Schreiber, and this mutation is lethal at a high temperature. So Drosophila normally live at 25 degrees Celsius. If you raise their temperature to 29 degrees Celsius, the individuals with this mutation will not survive. Another example is something we see in humans, PKU, which is a disorder of the metabolism of phenylalanine. So these individuals, when they metabolize the amino acid phenylalanine, which they get from their diet, it's converted into harmful products. And so what happens is individuals who are homozygous for this allele require a low phenylalanine diet, and therefore those harmful products do not build up and essentially changes the phenotype. An example of biological influence is something that we're very familiar with, pattern baldness. So heterozygous and homozygous males who have the allele that is associated with hair loss will experience a loss of hair. But only homozygous females will experience a loss of hair if they are homozygous for that same allele. So differences depending upon gender here. Something else we see in terms of the influence of the biological environment on genetic expression and phenotype expression is something called incomplete penetrance. And this is when individuals do not show a phenotype that you would predict based on what their genotype is. So a common example here is polydactyly, which is essentially extra fingers. So you see here a sixth finger on both hands here. And if you look down at this pedigree, this individual here, this male individual, is individual 3-2. This is a male, and his mother has polydactyly, and his child has polydactyly, but he doesn't. But you would assume that he would because his mother did and his child did. However, he does not. So that's incomplete penetrance. Another example of the influence of biological environment on genetic expression is something called variable expressivity, which is essentially very simply means that you get a variable phenotype even with the same genotype. So this is a great example shown here, the lobi mutation in Drosophila. Each of these organisms, each of these individuals seen here have the same genotype. They're all heterozygous for the lobi mutation, but if you look at their phenotype, they vary from individual to individual. This is variable expressivity. Another example of how the biological environment is going to influence phenotype is something called gene interactions, and this is a common phenomenon and something that we can illustrate by looking at the phenotype of chickens. So the idea of gene interactions is that one trait can be determined by two different independently assorting genes, and that different combinations of these alleles from two different genes can result in different phenotypes. And this is due to the interaction of the gene products with the proteins in the cell. So these two different genes, these alleles, are going to make different proteins, and the way those different proteins interact with each other is going to produce different phenotypes. So for example, this chicken comb shape, in these chickens here, you have two genes, the R gene and the P gene. Each of them has two alleles. So there's a dominant and recessive for the R gene and a dominant and recessive allele for the P gene. If you have a dominant R, either homozygous or heterozygous, this dash here means it could be the homozygous or the dominant or the recessive allele. And then if you're homozygous recessive for the P allele, you get the rows shown here. Homozygous recessive R with a dominant P gives you the P phenotype shown here. Two dominant alleles, one for R, one for P, gives you the walnut shape, and homozygous recessive for both produces a single comb shape. And this next slide just essentially repeats what was shown in the previous slide, just showing you what you get if you were to mate two different chickens together and then mate the hybrids to each other, and just showing you in this Punnett square, just showing you how these different phenotypes will arise. Okay, yet another example of the biological environment and its influence on phenotype, something called epistasis. Now, epistasis is defined as whenever a trait is determined by two or more genes, an allele of one of them can override the effect of the other on the phenotype. So an example, going back to Drosophila eye color, we've seen a lot with cinnabar in this chapter. So keeping in mind that the cinnabar mutation produces bright red eyes. However, the white mutation produces white eyes. But both mutations together, so flies that have both mutations, will actually have white eyes. And the reason for this is because the white mutation is epistatic to the cinnabar mutation. So the white mutation essentially overrides the effects of the cinnabar mutation. So why? Why does that happen? Well, essentially the cinnabar mutation is what's producing the pigment, but the white mutation has to do with the transportation of the pigment protein to the eye. So if you have a mutation, if you have a white mutation, you can't transport any pigment that's made to the eye. So even if the cinnabar mutation so with the cinnabar mutation there, even though you'll get bright red pigment, the white mutation cannot deliver it. The white mutation means that the pigment cannot be delivered to the eye, and so you're going to have white eyes. All right, another example of epistasis. These are sweet pea plants, and you can see they normally come in two different flower colors here. Now, the flower colors are determined due to a pigment called anthocyanin. So these purple flowers have anthocyanin. The white flowers don't have any pigment at all. Now, individuals that have two dominant alleles from two different genes, okay, so this is, again, because we're talking about epistasis, we're talking about two different genes, they will have purple flowers. But the homozygous recessive condition for both genes actually has the same phenotype as being homozygous recessive for only one and having a dominant allele for the other, or being homozygous recessive for the other and having the other dominant allele. So what's going on here is that the dominant allele is needed for a step in the production of this anthocyanin molecule. And each of the recessive alleles is epistatic over the dominant allele. All right, so what do I mean by that? If you have a dominant allele for the C or the P gene, okay, you're going to get anthocyanin production. And the reason for that is because anthocyanin, that molecule, is made from one molecule called the precursor, which is converted into some molecular intermediate using C as the enzyme that catalyzes that reaction. This intermediate is converted into the anthocyanin product by this second enzyme, the P enzyme. So as long as you have a C and a P, these dominant alleles, you can make anthocyanin. However, if either one of these are missing, so whether you have one condition of homozygous recessive here for the P allele or here for the C allele, or both of them being homozygous recessive, either way you cannot get that final product being produced, and you will end up with white flowers. One last example of the influence of the biological environment on phenotype is something called pleiotropy. And a gene is said to be pleiotropic if it affects many phenotypes. So just like we can have one phenotype being affected by many genes, we can have one gene that affects many phenotypes. So for example, back to PKU, gene mutations in humans, and that the accumulation of those harmful end products from that metabolism of phenylalanine can lead to many different effects. These people have mental impairment. It changes hair color, have a light hair color, and then also the presence of metabolites are found in the blood and the urine. And these are all phenotypes, all characteristics or traits that are determined by or controlled by one allele. Another example, back to Drosophila, the singed mutations. So there are bristles that are present on the head and thorax of Drosophila. The singed mutation not only affects the shape of these bristles, but also egg production. Okay, this concludes part 2 of chapter 4. Join me for our next installment looking at section 3.